Ribosomes are molecular machines that read processed mRNA and translate it into amino acids, the basic building blocks of proteins. Ribosomes are about 30 nanometers in diameter and have two subunits, a small and a large subunit, each with distinct functions. The subunits are made of about one-half RNA and one-half protein in eukaryotic ribosomes. Ribosomes are either found free in the cytoplasm, in the mitochondrial matrix, or bound to the rough endoplasmic reticulum. For translation of mRNA to occur, something must bring in the right amino acids based on the mRNA sequence. tRNA are molecules of RNA with an amino acid at one end and a specific sequence of 3 nucleotides at the other end called an anticodon. The anticodon matches the opposite group of 3 nucleotides, called the codon, on the mRNA being translated. The first step of translation is called initiation, where initiation factors guide an mRNA to the small subunit. The first tRNA attaches to the initiation sequence, and then the large subunit binds. The main step of translation is called elongation, where the tRNA enter the ribosome and attach their amino acids to the protein. Within the ribosome, there are 3 sites for tRNA. The ribosome will translocate one codon at a time to cycle tRNA through the different sites. At the A site, tRNA is checked to see if it matches the codon and it binds. At the P site, the tRNA donates its amino acid to the growing protein. At the E site, the empty tRNA leaves the ribosome. The last step of translation is called termination. At the end of the mRNA, the ribosome reads a stop codon, triggering release factors that halt protein formation and cause the subunits to dissociate. The subunits are either reused to form another ribosome or are broken down. Protein translation is critical to life, so ribosomes make very few mistakes. This is due to the highly specific anticodon-codon binding and other supplementary mechanisms. There are 4 different nucleotides in RNA, A, U, C, and G, and 3 nucleotides per codon. So there are 64 potential different codons. However, only 20 common amino acids exist, so multiple codons mean the same amino acid. This redundancy decreases mistakes during translation. When a tRNA anticodon matches up with a codon, the first 2 positions must pair exactly. However, the third position is less specific and is known as the wobble base. This would cause problems when trying to translate the AUA codon for isoleucine, but cells have evolved enzymes called synthetases that modify tRNA anticodons to prevent errors.